Hello my dear students and welcome back to Excellence Batch and I am your Diksha ma'am. So here we have started the chapter Biotechnology and its application, and so far we have done the application of biotechnology in the field of agriculture, in the medicine. We have seen how insulin is produced. Now it's time to see what are the other application of biotechnology in medicine and also in transgenic animals. So let's get started. So, gene therapy. The next application of a biotechnology in medicine is gene therapy. What is gene therapy? Therapy means to find the cure of something, right? You must have heard of it. And gene word is included. So, something it is related with the gene. Gene therapy is basically a process by which you cure genetic disorders. Right? What are genetic disorders? Inherited disorders that can pass from one generation to another, and they're controlled by genes. If any copy of a gene is defective in a baby, that can lead to a genetic disorder. Right? So they're present since birth and they passes from generation to generation. So you just can't control it. They can just come during the crossing overs and so fine. So how can you cure them? So for that, biotechnology have an option, and that's gene therapy. So if I talk about the definition of gene therapy, this is basically gene therapy is used to correct genetic disorder by adding correct copy of gene. By adding correct copy of gene. So if someone is having a genetic disorder, that means a copy of gene is defective. And what if we give them the correct copy of gene? For example, if a person's body, if a baby is born and baby is born with the wrong or defective copy of gene, why not to change it with the right one? So yes, that's what gene therapy is. So the first gene therapy was performed in 1990. Which year? In 1990, in a 4-year-old girl. They performed first gene therapy. So this girl was suffering from a disease, and the name of the disease was SCID. Severe Combined Immunodeficiency Disorder. So SCID was a disease by which this baby girl was suffering, right? So what causes SCID? So in your body you have bone, right? You have bone. So in the bone you have red bone marrow. You have red bone marrow, and in this red bone marrow, all the blood cells, they are formed and produced, right? So imagine these are all the cells that are getting matured and proliferated. Fine. So what do we want? We want that if you want these cells, imagine these cells are your lymphocyte. Which cells are these? Lymphocytes. The WBC, a type of WBC. And which lymphocyte? T lymphocyte. These T lymphocyte, if they want to get mature, if they want to get formed, if they want to get proliferated, we need one enzyme for their proliferation. And the name of the enzyme is ADA. What is the enzyme? ADA. Adenosine deaminase. Right. This enzyme is essential for what? Proliferation of T cell. Proliferation means the maturation and the multiplication, right? T cells are what? These T lymphocyte. Okay. If this ADA is absent, imagine there is a gene that secretes ADA. Imagine this gene which is secreting ADA is defective. Then ADA will not be secreted and T cell will not be formed properly, right? And if T cells are not formed properly, the person's defense system is not working properly, because T cells are very important in your immune responses. If T cell is not working properly, then The person's immune system will go down, leading to this disease known as SCID. So in SCID, a person's immune system is not functioning properly. As a result, the person can catch a lot of another diseases like tuberculosis, pneumonia, and so, right? Why? Because defense system is not working properly. Okay, so this enzymes copy ADA deficiencies there in this disease. And if there is ADA deficiency, no T cell proliferation. If the T cell does not proliferate, there will be immune system deficiency. So that was a problem. Now, what are the methods by which you can cure this disease? Let's see what are the treatment options we have for this disorder. What are the treatment options we have? The first treatment option we have is bone marrow transplant, because these cells, they are getting proliferated in the bone marrow. As I've told you, it's a red bone marrow. You can see here this structure, what it was, red bone marrow. Okay, so you know all the blood cells are formed in red bone marrow. So one option that can be, if a baby is born, so we can change the bone marrow. But is it very simple to take a bone marrow? No, it's not, because first of all, you need donors for that. Second thing, your tissue should match. So it's a very less probability that your tissues will match with the appropriate donor. So that's the biggest problem, and that's very expensive. Who will give the bone marrow? No one, right? Second is ERT, enzyme replacement therapy or treatment. So what we do in this one, because the ADA is not sufficient, why not to give the injection of ADA enzyme? You produce the enzyme artificially and you inject it. But again, this is not a permanent solution. Why? Because you have to give the enzyme after days or week after weeks, right? So if a person does not receive it, maybe the person will again start getting the symptoms of SCID. So that's also not a permanent cure. The third is gene therapy. So gene therapy can only be a permanent cure if you do it in the embryonic stages. So it's a permanent cure, not in the adult, not after a baby is born, but permanent cure if done in embryonic stages. Now why? Let me explain you how the gene therapy is performed, then you will get to know. For example, a baby who has been born, or a person who is born, or let's take an example, this 4-year girl child on which gene therapy was performed. Okay, so the person, imagine this is a baby who is born. Okay, so what they do, from the blood of this person, they take out the lymphocytes. What do they take out? They take out the lymphocyte. What are these? Lymphocyte. So from the patient's blood, they take the lymphocytes out. Okay. Now what they will do, once they take it out, they will culture it and grow it more in number. They will culture it and grow in number. They are increasing their number. Okay. So what they do? Culture of lymphocyte. Culture means you are growing them in artificial condition and increasing their number. Okay. Once they have grown a lot, what you will do? Now you will take a retrovirus. Okay. Now you will take a retrovirus and ask this retrovirus to infect a DNA in it. Retrovirus genome is RNA, right? Imagine this is a lymphocyte and this is your retrovirus. Okay, and you ask the retrovirus to infect this lymphocyte. And you all know that the genome of the retrovirus is RNA, not DNA, right? So, but we need gene, we don't need RNA. So how are we asking this retrovirus to put gene? Because retroviruses have a habit that their RNA, when it goes inside our body, it gets converted into DNA, and that DNA which is formed from RNA is known as cDNA. So technically we are asking the retrovirus to put cDNA inside the lymphocyte, and that DNA is what? Nothing but the correct copy of gene that is producing ADA, adenosine deaminase enzyme. Fine, you got it. So what is this? This is a retrovirus. Alright, so whenever we have to do the transformation of the animal cells, whenever we want that the piece of DNA should go into the animal cells, you cannot use plasmid because we are not bacteria. We want retroviruses to infect us and take it and give the copy of the DNA. So, but retroviruses have RNA as a genome. So, this RNA will first go inside our cell, and this will then convert it into a piece of DNA, and the name of the DNA is cDNA. So this cDNA is a DNA. C means complementary. Is a DNA that is formed from the RNA. So this cDNA is injected inside the lymphocytes. Now similarly, all the lymphocytes will be carrying this cDNA. So everyone have this cDNA. Why? Because retroviruses have infected all. Okay, now these cells, because they have the cDNA, they will be given back to the patient. So there is a benefit. What is a benefit? Because you have taken the cells from the patient's blood only, so there will be no rejection chances. The body's immune system will not reject it because that's the cells of the patient's own body. But the drawback is that these cells, they can die. They are mortal. These cells are mortal. They can die, right? So this is how we say that it can only be a permanent cure if you do it in the embryonic stages, because in embryonic stages, if you do in the early embryonic stages, in embryonic, which stage is early, when your embryo is only few cells small, at that time, because the baby is developing, all the cells are like stem cells. They will take the DNA and the entire body will get the entire DNA. Here, only because you're performing it on a born child, right, 4-year-old. So here the cells will only go to the person's particular body part. But if you'll do in the embryonic stages, every cell of the body will be having that DNA. Right? Every cell of the body will be having the DNA. So that's why we say it is a permanent cure. But if you do it in the afterbirth, then it will never be a permanent cure. Because then you take from the blood and put into the blood, but the entire cells does not produce any kind of ADA. Right? But if you do in the embryonic stages, the body cells, entire body cells will be having the cDNA. Okay? Or the correct copy of gene. So let's read few lines of NCERT, because this paragraph is very quite typical. You don't understand that. As a first step towards gene therapy, lymphocytes from the blood of a patient are grown in a culture outside the body. So as I've told you, we grow the cells outside the body. We take it from the blood. A functional ADA cDNA using a retroviral vector is then introduced into these lymphocytes, which are subsequently returned to the patient. Then what we do? We take the retrovirus, put the cDNA inside the cell, and then put the cells back into the patient. Right? However, as these cells are not immortal means mortal. Okay? The patient requires periodic infusion of such genetically engineered lymphocytes. So because they will die eventually. So you have to put them again and again. You have to do this procedure again and again. So that's not a permanent cure. However, if this gene isolate from bone marrow cell producing ADA is introduced into the cells at early embryonic stages, it could be a permanent cure. But if you do in the early embryonic stages, maybe the baby will be formed normal. Okay. Alright, so let's solve some questions. The first clinical gene therapy was given for treating diabetes mellitus, chickenpox, rheumatoid arthritis, ADA deficiency. Very simple, answer 4. It was given in ADA deficiency. Which of the following is commonly used as a vector for introducing DNA fragment in human lymphocyte? So what vector have we used? The option A is correct, retrovirus. Next, ADA is an enzyme which is deficient in a genetic disorder KIT. What is the full form of ADA? Very simple, adenosine deaminase, option 2. Okay, next, which kind of therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency? What therapy was there? Not immuno, not radiation, gene therapy. Radiation, immunochemotherapy are given in cancer patients. In gene therapy to treat adenosine deaminase deficiency disorder, which of the patient blood cells are used? So which cells have we used? We have used lymphocyte because they are responsible for immune response. Erythrocytes have no role in the immune response. Next, Next up, molecular diagnosis. Now, what are diagnoses? Yes. Okay. Diagnosis is the act or process to find out the reason and causative agent of disease. Right? Basically, we are just finding out everything about disease, what disease you have, and what is the causative agent of a disease. That's what molecular diagnosis is. Okay, that's what diagnosis is, not molecular diagnosis. That what diagnosis is. So it's a process by which you find out, first of all, what is a disease and what is the reason of disease. Two things. In diagnosis, what you find out, which disease is it, and reason of disease, or the causative agent of disease. All right. Now, if we talk about diagnosis, the diagnosis are of two types. One are the traditional method of diagnosis, also known as conventional method, and second are the modern diagnosis techniques. Modern diagnosis. So from like since childhood, you must have heard that if there is any elder person in your house, your grandmother, grandfather, or even your parents, right? You must have heard that if they are suffering from disease or just for regular checkup, they go for clinical laboratories for blood test, right? So from since childhood we are listening, do the blood test, you will get to know the disease. So that's a traditional or the conventional method. So in these method, basically what we take blood or or serum or urine, and then we analyze it, but these methods are always applied whenever you start getting the symptom of a disease. For example, if a person is suffering from maybe urinary tract infection, then person will not be able to urinate normally easily. There will be a painful urination. Okay, so now the person will go to washroom and find out that in the that in the urine there were blood clots. So now the person will go to doctor will ask for the urine test, and when the urine test will occur, urine analysis will take place. They will see the urine sample, they will find out what bacteria or yeast or any infection is there. Okay, so that means you are diagnosing a disease after the disease has caused to you. You cannot detect it before the disease has caused or going to get caused to you. So basically this is a method that you perform after you get the symptoms of a disease. The early diagnosis in this is not possible. For example, if I want to know whether I will have diabetes in what I say in later stages or not, or whether if I'll have X genetic disorder, I cannot do it with the blood analysis. Anyways, I have to go for the modern diagnosis. In modern diagnosis, early diagnosis is possible. Even the baby is not even born, I can still check whether the baby can have any genetic disorder in the future or not, right? So these include the tests like PCR, recombinant DNA technology. You have done PCR, you have done recombinant DNA technology. Yes, you also have heard of ELISA, and a new thing that we will study is autoradiography. All these techniques are new techniques, and these all are techniques of biotechnology. They come under modern diagnosis, and by which you can check whether a person can have any genetic disorder or any disorder later on in life or not. And this is what we call it as early diagnosis, right? So modern diagnosis involve all these techniques. Let's talk about ELISA first. ELISA. What is ELISA? Enzyme linked immunosorbent assay. So this is a technique. In this technique, what we do, so this we have done in the HIV also, if you remember. We take a plate. On this plate is attached an antigen. Imagine this is the antigen of HIV. Right, we want to check whether a person is having HIV or not. Okay, this is the antigen. So I will take the blood of the patient, and if the person is having HIV, the virus is inside the person's body, and that person's body will be producing antibody against HIV. So that means if a person is HIV positive, then person's antibody will be present, in person's blood the antibody of HIV will be present, and then it will join with this antigen. Antigen is already present on the plate, and this antibody is from patient's blood. Antibody from patient's blood, because we are assuming the person is HIV positive. Okay, now we have made one more antibody against the antibody of a patient. So this antibody is linked with the enzyme. So when you will add a substrate which does not have any color, it will meet with this enzyme, it will get converted into product, and the color of the product will be purple orange. So if you will find any purple orange color in your this plate, that means a person is HIV positive. Fine. So basically in ELISA, just with the help of antigen and antibody interactions, this is antigen, this is antibody interaction, we can find out whether a person is having any disease or not, or whether a person later on can get AIDS or not, because there is always a time frame between being HIV positive and having AIDS. So this is also one of the early diagnosis. Okay, so what if a person is not positive? Then this antibody will not be present in a person's blood. This antibody will not attach. If this is not present, this will not get attached. If this is not present, then substrate will never get converted into product, and purple orange color will not be seen, and hence person will be HIV negative. So basically in ELISA we do nothing, but we have some antibodies enzyme linked, and we have the antibody antigen interaction that help us to confirm the presence of the virus. Okay, all right. So the second diagnosis technique is autoradiography. Autoradiography. What basically is autoradiography? It is a technique. It is a technique to detect radioactive isotope in a biological sample. So if I give you any cell, a sample of cell, and I ask you, please confirm if this sample has any radioactive material or not. So the technique that you will use will be the autoradiography. Now how this technique of some radioactive sample detection help us to know genetic disorder? Let's see. For example, if imagine I'm a doctor, okay, or imagine to a doctor a patient arrive, right, and patient shows tells the doctor, yeah, these are the problems I am facing. Now doctor, by that knowing the symptoms and all and checking the genetic history of the patient, like mother and father, what disease they have, and so the doctor find out maybe the person is having a genetic disorder. So doctor want to check whether it is a genetic disorder or not. So what doctor will do? Doctor will ask for the test. So what happen in a test? In this test, we take the cell samples of a person. Okay, and in cell we have DNA, and in DNA we have gene. Imagine the doctor is thinking that a person is having muscular dystrophy. Okay, a genetic disorder. In muscular dystrophy, the person does not produce, a person's muscle cells does not produce dystrophin protein. If there is no dystrophin protein, imagine if there is no dystrophin protein, the muscle paralysis can take place. So imagine AT. This is a gene. This is a gene that produces a normal protein. Okay, If a person is having this gene, that means a person is normal and does not have any defect. Imagine in a genetic disorder via mutation, maybe this two nucleotides they get removed and a person gets genetic disorder. Okay, a deletion took place and a person have genetic disorder now. Okay, there is a difference. So this is a gene which is mutated. And mutated gene causes formation of abnormal protein or no protein, and hence that's a genetic disorder. Alright, so if a person is normal, the person might be having this kind of a genes. If a person is having disease, the person may have such kind of a gene. Okay, so now what we do, we use a probe. What is a probe? A probe is any RNA or DNA with radioactive isotope, radioactive phosphate, and the phosphate uses P32. Okay, the radioactive phosphate is used. You all know in our DNA, the backbone is made up of sugar phosphate bonds, right? So the phosphate now we are using in this piece of DNA known as a probe is radioactive phosphate. So we have two conditions. Condition number one, if a person is normal. Condition number two, if a person is having disease. Okay, so we are first will take about this one. Imagine we took a sample of a person and we took the gene because we think maybe this gene is defective. Now we will ask a radioactive probe, which is the radioactive, which have radioactive phosphate, and we allow it to bind with the piece of DNA. Okay, because the person's gene is normal, this probe can easily attach to it because it is complementary to this. So we use probe in such a way that it will combine with this one, right? Because we know normal gene is this one. What will be a mutated gene? You have no idea. We are assuming maybe the deletion has taken place, maybe the mutation is in another way. You cannot assume what kind of mutation has taken place. But one thing that you can assume, if a person is normal, the person will be having such kind of a sequence of DNA. Right? Now, if a person is normal, the radioactive probe will attach to its sequence of DNA. Right? And once this attached to the DNA, if I keep it on a photographic plate, I put some light, I can see the light emerges out from this photographic plate. So if a light emerges out, that means there is a presence of radioactive material. And the technique that I'm using, putting it on a machine, photographic plate, and then I give some energy or a light, and a light emerges out. I can see the emitting of light. That means a person's genes are normal. So this technique is a autoradiography. Okay. But what if a person is having a genetic disorder? If a person is having a genetic disorder, this probe cannot attach. The probe cannot attach. Why? Because you all know how the probe is. The probe that we have made is TA, T, 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 A. Can A bind with the A? No, it cannot. So this probe cannot attach. If it cannot attach, there will be no emitting of light during autoradiography. So when you will see there is no light on a photographic plate, then that means a person is having genetic disorder. If there is a light, that means a person is quite normal. So this technique is also used to check the genetic disorders. Okay, so these were the diagnostic techniques. Let's solve this question. Which technique helped to identify a bacterial or viral pathogen in a human body even when its concentration is very low and clinical symptoms are not yet visible? Differential leukocyte count, ELISA, total leukocyte count, and PCR. DLC, TLC is what? It's the count of leukocyte, that how much lymphocyte, how much WBCs are present in your blood. Total is total leukocyte count. Differential is the count of different types of WBCs in your body. For example, how much is your or how many are your eosinophils, what's the percentage of your lymphocyte, what's the percentage of neutrophil, and they usually done with the blood analysis, so it's a conventional method. In ELISA we do antigen antibody interaction, but PCR is a technique that helps in the amplification of DNA. We have done in the previous lecture. So whenever you have any DNA material in a low quantity, what technique will you use? PCR. So answer to this question is 4, right? Okay. Now let's talk about transgenic animals. Now what are transgenic animals? Transgenic animals are animals that have their DNA manipulated. Right, DNA manipulated means either some gene will be added, either a gene will be subtracted. So we have an abundant of transgenic animals, like we have sheep, we have cow, we have monkey, we have spiders, right, and we have mice. So more than 90% of your transgenic animals, I think more than 95% of your transgenic animals, they are mice, right? So this is one example. You can see this is a very small mice, this is the larger one. So what we have added in this one, this mouse is known as super mouse or super mice. Super mice is a transgenic mice in which we have added growth hormone gene. GH means growth hormone. So here in the super mice, there is excessive formation of growth hormone that will lead to the excessive growth of the mice. So normal mice is this size, but this transgenic mice is of the larger size. Fine. So this is one example of how you have manipulated the animals. There are number of examples. Let's start this one by one. So let's see what are the other animals that have been made transgenic. So first of all, we'll talk about sheep. So in sheep, the first example that we have is Dolly, Polly and Molly. So Polly, Molly, Dolly all were Eves, right? Polly and Molly, these were cloned and transgenic sheep at the same time. So these were the first transgenic sheep that was formed, right? Whereas Dolly was the first clone that was formed. What's the difference between being a clone and being transgenic? Clone is something that is formed from somatic cell. For example, if you take my hand cell or the cell from my hand and make a baby out of it. So that's what clone is. Transgenic is that now from my cell, this cell, you are taking the cell also and you are also manipulating the gene of it. That is clone and transgenic. Okay, so Dolly was a sheep which was the first clone and this was clone and transgenic. Now these were produced at an institute, that is Roslin Institute, Edinburgh, that's in Scotland. Okay, the second sheep that we have is Tracy. So Tracy was also a sheep and that was a transgenic sheep. That was a transgenic sheep, right? So Tracy contains a gene. Tracy have gene that produces alpha antitrypsin gene. So what this alpha antitrypsin was used for? So this was used for a disease known as emphysema. You all must have heard of emphysema in your class 11's breathing and exchange of gases. Emphysema is a disease that occurs due to excessive cigarette smoking, right? When a person smokes, what happens to its respiratory tract's wall? When a smoke enters inside the person's respiratory tract, what happens? So imagine the smoke enters inside the respiratory tract. What is it? Respiratory tract. Okay. Respiratory tract. Now imagine the smoke enters in the respiratory tract and smoke have certain chemicals. Right, so these are smoke chemicals. So what these smoke chemicals can do inside the wall of your respiratory tract, it can activate an enzyme and the name of the enzyme is trypsin. Once this trypsin is activated, because it is protein digesting enzyme, it can break all the fibers, elastic fibers of your respiratory tract, and that causes loss of elasticity in the emphysema. So antitrypsin, alpha antitrypsin is the one that will block this trypsin, and hence it is used for a disorder emphysema. Fine. So what is the role of alpha antitrypsin? It blocks trypsin enzyme. Trypsin enzyme. Fine. So this is another sheep, Tracy, which is very important. Please put a star on it. That is transgenic and has been used to cure a disease. Apart from that, we have also used transgenic animals to treat phenylketonuria, okay, and other such diseases. So let's talk about a monkey now. Third is a monkey, and the name of the monkey is Andy. Andy is a monkey, transgenic monkey. Now what does this Andy have? Andy have a gene which is GFP gene. What is GFP? GFP is green fluorescent protein. So this green fluorescent protein is isolated from a jellyfish, and this is responsible for bioluminescence. Yes, so now Andy also shows bioluminescence. What is bioluminescence? Bioluminescence means anything that emits light, right? So the gene from the jellyfish is transferred into the monkey. Now monkey is also emitting light. Okay. And now fourth one is a cow, and the name of the cow is Rosie. So Rosie is a transgenic cow. So this cow secretes milk, because it's a cow, definitely it will secrete milk. It produces protein rich milk. So we have added a gene of a protein. Which protein? Alpha lactalbumin. So this milk is very good for the infants and the babies because it is rich in a protein. So we have added a gene which is alpha lactalbumin in a cow. Now cow's milk contain high quantity of protein. How much protein? 2.4 gram per liter of protein is present in a milk, right? So in 1 liter of a milk you can find around 2.4 gram of this protein. So it's a protein rich milk. Okay, so see, these transgenic animals have been used so far for producing products and so, right? Now let's see what is the application or the benefits of transgenic animals. Benefits of transgenic animals. The first benefit of a transgenic animal is in the medicine, in the field of medicine. Second benefit is in the field of agriculture. And third benefit is in the field of industries. Let's talk about them one by one. First, we'll talk about medicine. Now, what's the use of it in medicine? First of all, by making transgenic animals, you can find out that how a normal human physiology functions. And also you can find out how a disease occurs. For example, if there is a particular gene that is mutated in a disorder, you just can't do all these experiments on a human body. This is completely unethical. But if you make the same gene mutated in a mice, because mice is a mammal and near to us, then you can find out what actually happens during that disease. So by using transgenic animals, what you can see, you can study normal human physiology, that for example, normal human physiology. For example, you can find out that how our particular enzyme is secreted from a gene. That's a normal human physiology. And we can also study diseases, any kind of a disease, how a virus attack us, how COVID virus attack us, and everything can be studied by transgenic animals. Apart from that, you can also use them for biological products. The examples we have done so far is Tracy and Rosie. Tracy and Rosies have been modified in a way so that we can get our products from them. Okay, and we can also make the animals no, not just for these emphysema disease. As I've told you, we have also produced them for phenylketonuria and cystic fibrosis also. So for products we can also make transgenic animals. Next, for chemical testing, chemical or drug testing, whenever you make a drug, you just, for example, someone invented a drug against any disease, can you just directly give it to the human patients? No, maybe that has a lot of side effects. So first you try it on the transgenic animals, like on mice, right? And also chemical safety, for example, if you want to check if this is any product, does it secrete any toxins or not, and these toxins, if they can be harmful to the human body, they can be tested on mice. Okay, another thing is vaccine safety. So whenever a vaccine is produced, again you cannot try it on the human beings. Maybe the vaccine which is containing the virus, maybe that virus you are giving as a vaccine, but that causes a disease. So first you have to check it on the transgenic animals. So for all these studies, in medicines, the transgenic animals have a major role. Fine. Let's talk about agriculture. In agriculture, the first role is in breeding of animals. In the conventional breeding technique, what they used to do, they took a female and a male cow, like a bull, right? And this female is with the superior qualities like high milk production, and male was also superior, like disease resistant. And they were allowed to get cross and see how their babies are formed. Imagine they formed the babies. Now, what are the chances that all the babies will be having superior character? Very few. So conventional breeding technique is not like 100% successful. Might be you will get the character, maybe not. For example, sometime you have seen both the parents, they are tall, but their children, they don't have very taller height. So that's a kind of an example that not always the crossing over that will take place in normal sexual reproduction will be successful with the good genes. Fine. So if we use this technology, biotechnology, the success rate is more. Why? Because you will manipulate the gene. Imagine you directly put gene in the embryo for taller height. The baby will be taller, right? Or you put a gene, right? Or you manipulate the ovum and you added a gene that it will be containing a gene for high milk yielding quality. Definitely the baby will be having high milk yielding quality, right? So that comes to the second point. That means to increase the quality of animals or their products. For example, Rosie was a cow. The cow was producing a lot of milk, and the good thing was the milk was having high protein value, right? So that was how we have increased the quality also. If we make some fishes manipulated with the genes, maybe the fishes will have more omega-3 fatty acids and we can have them by eating them. Okay, so this is how we can also apply that in agriculture. Agriculture doesn't mean that it's about plants only, it's also about animals. Animal husbandry, we'll be doing this chapter after this. Okay, all right. So these are some of the application in the agriculture, that how you can use this organism, and also we can make our organisms disease resistant, right? We can also make them disease resistant by manipulating their genes. That means they will have a strong immunity, and just little microbes, they can't do anything of them. So all these are the benefits of transgenic animals in agriculture. Now let's talk about industry. Okay, now ma'am, what's in the industry? Let me give you an example. In 2001, Nexia Biotech, it's a company in Canada. They have taken a spider gene and they added that in lactating goat that was producing milk. Now the milk have fibers. Which fibers? The gene that we use of the spider was web gene, and the web that a spider make is equivalent to the silk thread, So all these fibers, what they did, they isolated and they used them to make rackets. Which racket? Tennis racket. Yes, even they made sutures that you get when you get injury, right? Doctor give you sutures. And also this was used to make army uniform, because these fibers, they were great in tensile strength, they had a lot of strength, that they have been weaved and used to make products. So this is how this can also be used in the industry. But somehow this is not good, because you are, what are you doing? You are making these animals, you're just misusing these animals basically. Okay, and that's not good. So here come what is good, what is not. How will we know that the thing that we are doing, is it good for the nature or not? Is it good for the animals or not? So here comes the ethical issues. So we have a term in our biological field, that is bioethics. What are bioethics? Bioethics are the set of standards. They are set of standards in biological world. In biological world, to have control on inventions and discoveries. Right? For example, if we will just allow every scientist to make whatever they want to make, then something bad can be made. For example, we say under these rules of bioethics, you cannot perform any experiments on human. So that's why there is no experiment directly done on the human. If you want to give a drug, if you want to invent a drug, you have to pass through set of first animals. If they are successful on those animals, only then it can be given to the patients. Okay. Now, not again, it can not directly given to the patient. First, there will be group of people who can have the drug, and then it will be sent to the others. Okay. So this is how we have certain rules and standards, so that you can do whatever invention you want to do in a confined circle. You cannot go out of it. So if if any scientists want to do any research, they have to go through certain qualifications, and they have to go for certain permissions and all. After they got all the permissions from certain committees, only then they can do that research. Because there can be a thing, for example, while doing research, you produced or you manipulated a virus, and that virus causes a disease that can be havoc, right? And that can destroy the human race, and even you don't have the a particular treatment for that also. So to avoid all these circumstances, there are certain rules. So for example, in India, we have a committee, GEAC, Genetic Engineering Approval Committee. So these things are controlled by these committees, and they will make decision which research is good, which is bad, which should be done and which should not be done. If for example, if you want to do research, the research will go to them. If they find that yeah, this is a good research, they will give you permission, you can do it. If they find out there is something fishy, then they will not allow you to do that. Okay. So doing something which is against the bioethics is a crime, and scientists can go to the jails and all, right? So these are bioethics. And next we have is the biopatent. Now what is a biopatent? After you have made any invention, you can have certain rights of that invention, so that no one else can claim to it. For example, I produce a particular drug on a particular disease, and there was no one who have ever produced any drug on that disease, right? So this is a kind of a novel thing. Novel means new thing. So I can get a set of right that I have a right on this drug. Whatever I want to do with the drug, that is my choice. Okay. I can also give some of the right to the pharmaceutical company. They can make the drug, and I can take a share, or I will not share with any of them, or I can produce the drug and then share with other people from my hand only. So that are the rights that a person have, and that is known as biopatent. So biopatent are nothing but these are rights given by government or any agency to inventor or assignee of an invention or discovery. If you have even made any discovery, even then you can get a patent of it. Okay. So these are given so that you can disclose with the public and relations, and no one else can claim to it. Okay. So Indian Parliament has also passed a bill there, right? Indian Patents Bill for for these patents, bio patent, so that if there is a conflict, you can go to the court and you can fight for that. And second amendment has taken place of this patent bill, and this is for the that takes place such issues into consideration like patent terms, emergency provisions, research and developmental initiative. So all these things they cover under Indian patent bill. Patent is very important. If you don't take a patent, other people can get claim to it. Okay, so if for example you don't have a patent of it, another takes the claim to it, then that's a case of biopiracy. For example, I invented something but I don't have a patent right now, and some other person from other country, they saw my research and they find out I don't have patent and they took patent of it. So that's that's a crime, and you call it as biopiracy. So what is biopiracy? It's the term used to refer to use of bioresources by multinational companies and other organizations without proper authorization from the country and people concerned, without compensatory payment. So some multinational companies, they come from other countries to the countries like India and Africa, who are very rich in the culture, who are very old in their civilization, who are rich in the bioresources. They will see any of our plant. For example, they will find out in India it is said turmeric have anti-inflammatory properties. That's why since time immemorial we are drinking turmeric milk whenever we get any injury or so, right? But we don't have any patent of it many times back, right? So we don't have a patent of it. So what they did, the US university searched on turmeric and took a patent of it. So that was a crime, because in our Vedas everything is written clearly. If we don't have a patent, doesn't mean that doesn't belong to us. Okay, so such kind of things are the crime and you can't do it. Such kind of a crime was done in 1997 when an American company known as Rice Tech, the name of the company was Rice Tech. This was a company in Texas. What they did, they took their semi-dwarf variety, which was their rice, and they crossed it with our basmati rice. Everyone in the world knows basmati belongs to India. In India, we have around 2 lakh rice varieties. More than 2 lakh, out of which 27 are documented basmati. Right? So they took any of our basmati rice variety, they crossed it and they made a new rice plant. And they took patent of it. Right? So this is a crime. Why? Because you didn't ask us. You took our basmati rice variety, you crossed it and make a new one, but you didn't ask us. And even you took a patent on it. How can this be possible? Right? So later in years, you will then say, don't eat basmati, give us first money because we have rights on it. So this is completely unethical. In our Vedas, in our books, in our folklores, everything is written about rice. How can you take it? So this case was sent to the WTO, World Trade Organization, and some of their rights were taken back. Same was done on turmeric and neem. The turmeric right was taken by US University of Mississippi. Right. Similarly, the neem's biopatent was taken by Germany people. Right. W.R. Grace and Company. There was a company who took the right of neem, patent of neem. And what patent they have taken? The fungicidal property of neem. But we use neem face wash and they are telling us fungicidal properties of neem. Right. We are eating neem for a lot of our skin allergies and they are asking us. So these rights were also taken back, right? So this is a biopiracy. Piracy means a crime. Patent means a right, and ethic means the rules. So these certain rules and all these things, they are very important in biological field also. Whenever you are doing any research, so the government is also involved in that. Okay. All right. So let's solve some questions based on it. Use of bioresources by multinational companies and organizations without authorization from concerned country and its people is called. Very simple. Use of bioresources without authorization is a crime. That is a biopiracy. Okay, next. In India, the organization responsible for assessing the safety of introducing genetically modified organism for public uses. Simply, there is only one committee. I have told you, GEAC. This is a committee that see everything. Learn its full form also, because once in exam the full form was asked, right? Okay. Which body of government of India regulate GM research and safety introducing GM organism for public services? Again, same kind of a question. Answer 1, GEAC. The laws and rules to prevent unauthorized exploitation of bioresources is termed as rules and laws. They are bioethics. Rights is biopatent, and biopiracy is a crime, and bioengineering is the genetic engineering. Match the following: Bt cotton, adenosine deaminase deficiency, RNAi and PCR. Okay, so you can also get such kind of match the following question from both the chapters of biotechnology. Gene therapy, cellular defense, detection of HIV infection, Bacillus thuringiensis. So for first, Bt stands for Bacillus thuringiensis. So fourth, adenosine deaminase is an enzyme that is used to proliferate T cell and helps in the proliferation of T cell, and hence these enzyme, they are not formed during the genetic disorders kit, and hence we do gene therapy. 2-1. Third, RNAi is a cellular defense mechanism of your eukaryotes, and PCR helps in detection of HIV infection. So not just ELISA, but PCR is also the one we have done that it helps in detection of diseases. So for 1 we have 4, 2-1, for 3 we have 2, and this for 3 answer is 4. Okay, simple. So that's it about biotechnology and its application. So I will meet you in the next class. We'll be starting with another chapter, probably evolution or the strategies. Let's see. So just finish this up and I'll meet you in the next class. Bye-bye. All the best. Take care.